want to welcome our panelists on stage. That is Dr. Sarah Stelzsche, it's Professor Eva Hellström-Lindberg, Professor Malene Hillebrandt, and Professor Peter Nagy. Please come up here, and please, the rest of you, welcome them on stage. I don't mind where you sit, just as long as you're comfortable. All right. So, and I want to specify Professor Eva Hellström-Lindberg next to me from Karolinska Institute in Sweden, who's been advancing precision medicine approaches in blood cancers and myelodysplastic syndromes. And Dr. Sarah Stelzsche from the Center for Genomic Regulation in Barcelona, a pioneer in understanding chromatin metabolism and epigenetic vulnerabilities in cancer. Professor Malene Hillebrandt from Odense University Hospital has focused her research on utilizing the glucose metabolism of breast cancer metastases for guidance in oncologic monitoring and thus in treatment decisions. And we have lastly Professor Peter Nagy, National Institute of Oncology in Hungary. All right, so let's have a look at this at first. I guess diagnostics could have been the biggest word in any of the word clouds that we performed, but it wasn't, but it is now. Why do you think that is? Malene?
Yeah, I think it may be because of PET scans.
Yeah.
Using fluorodeoxyglucose,
Yeah.
where we apply, and it's based on this Warburg effect that cancer cells.
The audio gods are against us today.
That cancer cells have this higher glucose metabolism than normal cells.
Right.
And it's used a lot and applied a lot in diagnosis of cancers and staging cancers.
Mm-hmm. Mm-hmm. Dr. Nagy, what do you read into novel targets?
Well, that relates to the research that we're pursuing at my laboratory very much, because for a long time, people only thought about cancer treatment in terms of genetics and genomics, but now we know that these mutated genes, they actually translate into malfunctioning proteins, which are the bad guys. They're the ones that are responsible for the malignant transformations of the cell. So when we design a drug or utilize a drug, we always treat proteins, not genes. And now we also know that those proteins that needs to be targeted, which are the targets, needs to be very specifically characterized before we can elude into drug development endeavors. And we now also know that many proteins have impact on each other's functions. So protein-protein interactions are very important nowadays. And in metabolic rewiring, because that is the topic of today's panel, these malfunctioning proteins not necessarily need to be mutated. Sometimes it's enough if they are a little bit more abundant than they normally should be. And to better understand how they contribute to metabolic rewiring within the cancer cell, being it response to immune system related events or any type of treatment, is, I think, very important in order to be able to design effective drugs which have not severe
and you
side effects.
I think you actually answered my next question now, which was, what are you working on? And I'm going to get back to you and ask what are the prime challenges that you're facing.
I can tell you more about that.
Okay, I would like to ask you, Professor Hellström-Lindberg, what word did you write?
I wrote understanding.
Understanding what?
Understanding why a cancer cell expands and takes place in the bone marrow. And I work with hematology, and we have the luxury of being able to take out single cells from an endless resource, which is the bone marrow. And we know that in most cases, there are cancer stem cells. Sometimes they are actually the same as the blood system's stem cells.
Oh.
And we need to understand why they compete. And I would like to comment on something that you said, they grow uncontrolled.
Yeah.
But it doesn't have to be the case. They just have to, the stem cells of the cancer have to be a bit better than the normal stem cells.
Right.
And sometimes they do it because they divide more often.
Yeah.
And then we're not talking about super much often. Perhaps they divide 10 times per year instead of 8 times per year or 4 times per year. Sometimes that is the case. They actually have in the cell a growth advantage. But sometimes, and we know, for example now, I think not only in hematology but also in many other cancers, that the cancer stem cells, the mutated ones, can be there for decades before they actually emerge as a cancer. And then they may actually win, not because they actually are better, but because they do something to the microenvironment that they are resistant against, but that the healthy cells don't like at all. And then we come to the proteins that you were talking about. I mean, they shift the balance in a very, very subtle way.
Wow.
So this is, and we need to understand, because with each mutational combination we get different gene expression and epigenetic sort of features, and eventually novel proteins, or some proteins just disappear. So that's why I think understanding is my biggest challenge.
I think that's a wonderful word. Thank you. Dr. Stelter, what did you write here?
Well, I would choose cell biology. First of all, because I am a cell biologist,
Yes.
and second of all, because I believe that cell biology includes all the rest, right? Because without studying cell biology and understanding better what the cancer cell is doing, right, we cannot find the right targets, we cannot find biomarkers, we cannot improve detection, we cannot understand how cells adapt when we give them drugs, right? And then related to this, I want to also maybe spend some words regarding what Peter was saying. No? We are often design drugs for a protein that's in the cell actually may be doing different things depending on the interactors with which it is interacting, right? Or depending in the compartment in which it is localized, right? So most of the time drugs are used to kill the function of that particular enzyme, in the case of metabolic enzymes. But in fact, I believe that we know not very much about what those metabolic enzymes are doing. So we are talking about the Warburg effect, which is one of the first things that we started discussing in the panel here. This metabolism is occurring, which is needed to break down nutrients and produce other building blocks. It's happening in the cytoplasm. What we have found through mass spectrometry and imaging and also metabolomics is that actually the nucleus is a metabolic compartment. Metabolic enzymes are inside the nucleus. They can perform metabolic reaction there. And those metabolic reactions are then required for shaping the DNA. So how the cell interprets, the information that is coming from the DNA. And this means, of course, that the cancer cell can use this, metabolic machinery inside the nucleus to remodel the DNA.
So it can almost feed itself?
Exactly. So we had until now the idea that metabolism is, the last layer, is how cells react to something, is an adaptation. Rather, what we are saying is that metabolism can shape this adaptation by directly speaking to chromatin and telling chromatin and the DNA what to do as an output of information for the cells.
So this is something you can use to identify whether it's a cancer cell or not, but also to control.
Yeah, so what we do in the lab is to see whether we can use this information that we have of what metabolic enzymes are going in the nucleus in the cancer cell to exploit this information to identify new biomarkers, for instance, for specific cancer types. And also, of course, if we know that that particular metabolic enzyme is essential for the cancer cell to survive or adapt following a specific treatment, we can target the translocation of the enzyme to then kill the cancer cell specifically.
Thank you. So Melina, is this the same that you're staring at to monitor how a cancer is developing?
Well, I would not say it's exactly the same, but we utilize the Warburg effect also for this being based on this increased glucose metabolism by cancer cells. And we use this for diagnostics. And as we just heard from Dr. Stelzy, this was 100 years old, this Warburg effect, and then 50 years after, the fluorodeoxyglucose, which is a radioligand for imaging, for PET imaging, it was developed, and it was it can be used in PET scans to see the glucose metabolism in cancer lesions, and therefore we can use it for cancer diagnostics and cancer staging. And what we do is actually we do the clinical application. Since it's already many years old, it has come to clinical practice, and here we use it, these FDG PET scans to monitor treatment response in breast cancer patients. So patients having metastatic breast cancer, we know that the FDG PET is much more accurate than conventional imaging that is not based on function or metabolism, and therefore we hypothesize that using this for monitoring treatment response, this will result in a patient benefit because you can better guide the treatment and make the right treatment decisions if you see changes in cancer metabolism over time.
With the difference that this is on solid tumors, right?
This is on solid tumors, yes.
Right.
And our project, our Horizon Europe project, is based on metastatic breast cancer, so it's actually only breast cancer, but it's a widely used across cancers, this FDG PET, and also other types of PET scans that target proteins and not only this glucose metabolism, but this is just what our research project is about.
Okay, Professor Narji. So what you're focused on is redox biology. And you just opened up a little door to what that was all about. But based on what Sarah just told us, Dr. Stelzy just told us, how does it compare? Just if we use that framework.
Well, if you want to connect, I mean, the Warburg effect is related to redox biology very much indeed. So we know that rewiring the glycolytic pathway actually contributes to oxidant production inside our cells. And redox is something that is not always familiar to the cancer audience, but I'm sure that the people, in particular the ladies who are listening to us today, have heard about antioxidants because they're a big hype now in the cosmetic industry.
Yeah.
And the reason for that is because early discoveries in the field demonstrated that excessive oxidation can damage our cells. That contributes to aging and also to cancer development. But today, we know that this is not only something that is a side effect of malfunctioning cellular events, but redox biology is indeed very important for some vital cellular functions. So we have some proteins that are dedicated to produce oxidizing molecules, which have very important biological functions. And if they don't work properly, then they can contribute to cancer development. But more importantly, to the topic of today's panel, these reactive species are really important in rewiring the metabolic pathways that we are studying or researching in terms of new treatment development. And that is mainly the topic of what we're doing. And we are focusing on a very specific segment of redox biology. That is the transsulfuration pathway. That is a metabolic pathway. And this is interesting because in redox biology, the most important and most oxidant-sensitive amino acid is cysteine. That has a sulfur, and that is why. And cysteine is a non-essential amino acid, which means that we don't have to eat cysteine. We can produce it in our cells. And this metabolic pathway, the transsulfuration pathway, is responsible for producing cysteine from methionine, from another amino acid. And to put it bluntly, what we and others have discovered in the last 2 decades was that some of the enzymes that are involved in cysteine production have secondary functions which contribute to rewiring of different metabolic pathways via producing active and reactive sulfur species, we call them, which impact other protein functions. And we have noticed that this pathway is very plastic, and the cancer cells like to utilize it. It's a stress response element, as we call it. So, for example, it can respond to the immune system, it can respond to treatment, either radiotherapy or any types of drug treatment. And it's a very plastic metabolic pathway, which means that the cancer cells can really use it to their benefit depending on what the stimulus is. So in some cancers, it contributes to angiogenesis and protection against the immune system. In other cancers, it's responsible for metastatic development. And the most exciting angle that we're pursuing now is how cancer cells utilize these pathways to overcome drug effects, to drug resistance. It's really important there.
Okay, so what I gather here, and I want to say to you all, don't just start asking questions if you have questions for this panel. I'm sure you might have. You can start now, and I'll get the message pour in. So what I'm getting here is that we can get on cellular level, you can get insight, you can find the cellular vulnerabilities, you can find the metabolic vulnerabilities, you can monitor, you can visualize, you can image, you can even maybe kill the cancer cell in a laboratory. But how far away are we from treating cancer with this approach?
So I think we need to understand what is the driving cancer cell, which I think is different when you are working with solid tumors than when you're working with, for example, leukemias.
Yes.
And other blood cancers. And we need to kill it. We can kill 99.99% of the cells. And
as you said before.
It will look perfect for a moment, but if we haven't killed
that
or
one
attacked the stem cells, it will come back. That's why we have to change bone marrow to some patients, because you have to give another person's bone marrow. But I think it's important that we have to be better at finding the cancer-driving cells. And since they divide very, very rarely, you have to sort of find their vulnerabilities. It's often a combination between finding the vulnerabilities, giving novel drugs or old drugs that sort of has the possibility to target them, and then to make them exposed to the immune system. Because one talent of the cancer stem cell is that it could actually hide the receptors for the immune system. So they hide, they go under something, they take another cell over like a blanket, and then when they think, fine, they pop up again. So we have to target them through most likely several mechanisms. That's why they are, I mean, the best thing for cancer if you can cut it out by surgery, but we know that will not really cure more than a fraction of our patients. So we need to understand them both as immune targets, targets for novel treatment, and also realize when we have to give up and change, for example, bone marrow.
Anyone wants to add anything to this? I mean, we're always talking about, I mean, we can be very impressed about the revelations that happen in the lab, and yet it feels like we're always waiting for that treatment.
Could I say one thing? Because I think these new findings in many cancers, including hematology, that we have a huge window. For some of them, it's quicker, but for some, it's decades.
Yeah.
And they go, and that gives us a therapeutic window. So if we can find these stem cells, and if we understand how they...
Then we have solved the whole problem.
You have time. Perhaps you don't have to give very toxic treatment. Perhaps you can find the way of targeting them in a
So what
better
you're
way.
saying is if we find that,
Yeah.
we will solve the entire problem of cancer.
No. No. There will
Why
be
not?
many. Because for each cancer type, they have a specific tumor microenvironment. They have specific mutations. Some grow rapid, as we said. Some smolder on for ages. But if we can find the toolbox, perhaps we can, in the future, set up the right tools, including immunotherapy. And then I also believe in vaccine strategies, which is less toxic and less cumbersome than many of the new cellular therapies.
But I thought vaccine was taking something, a little part of the disease, and put into the body. That doesn't sound wise when it's a cancer
because
cell.
you can. I mean, I think Peter has done the same thing. I mean, tumor cells can produce something called neoantigens.
Oh.
This is their own proteins, and they have changed it. They don't exist in the natural, healthy body. And if you can find these, and if you can then address and educate the immune system to find them before they are, if they are billions and billions, you can't do anything. But if you can find them at a low level, you can perhaps find, for some cancers, a way of actually vaccinating against them.
So the problem is that most tumor cells are very clever. They know how to overcome treatments, and now we have really a wide variety of treatment options. As we learn more and more about cell biology, I also put cell biology down to the question. And what we see is that in most cases, the cancer cells can still learn how to overcome treatment efficacy. And to my mind, the only good cancer cell is a dead cancer cell. So what we need to do is to understand how to kill them all. So what they do, if I want to put it bluntly, is that when you start treating a cancer, then some of the cells, they just go to sleep. They go to a dormant phase. They don't proliferate. They just sit there and rewire themselves to learn how to overcome treatment. And once they learned how to do that, they start proliferating again. So we have to understand a little bit more about these sleeping so-called persister cells and learn how to kill them very efficiently. And I think that could be an interesting way forward in designing more efficient therapeutic interventions.
Maybe we should try a little rapid fire here and say, so what do we need to build this house of with the understanding we have today, and we want to reach to the point where we can actually cure these types of cancer we're talking about here. What does it require?
Collaborations.
Between who?
between us in the lab, between hospitals, accessibility to sample patients, in which we can actually demonstrate that what we see in a Petri dish is actually what's happening in the patient, right? Collaboration with funding agency, of course, that, you know, this collaboration, these synergy grants, let's say, that put together people for a common cause, I think are a good instrument, a good tool for tackling these big questions. Especially because most of the time what we see in our model systems in the lab may apply to one specific subtypes of one particular cancer, but so we need to understand how generalizable the discovery that we have made in the lab is across different type of cancer, across different type of cells within the same cancer, right? Because the cancer is not just clever in the sense that it adapts to the treatment, but within the cancer you have several types of cancer cells because of heterogeneity that is intrinsic of the tumor that would react differently and would respond differently and adapt differently to the treatment itself, right? So this complexity cannot be tackled by just one lab or one person. It has to be a group effort, of course, and I think this is what is needed to
And
create this
you're leading an
toolbox.
effort, and you're collaborating with.
Yeah, we are collaborating, of
already.
course. It's sometimes difficult, for instance, in our case, since we are a basic biology lab, to really find partners in the clinic or in that have more expertise in translational research. So this type of collaborations would make an impact on what we discovered and to, you know, translate what we find in the lab to the clinic in the future.
Do you guys want to put the QR code on again so that if people want to ask a question, they can scan the Slido? What do we need? What else do we need? We have collaborations between labs, hospitals, funding agents, and so on and so forth. I'm going to ask you after what's stopping you, but right now let's just build the house first and then talk about what's wrong with that afterwards. What else do we need?
I think we need collaboration, as you say, between competences.
Yes.
And
Different
I think
competencies.
different competences with full respect, and we need, and patient material. But
Which
we need,
competencies?
I would say, at least for the diseases that I work with, you need hematopoiesis. You need to understand the problem that is you can cure leukemia. The problem is that you then also kill the patient because you can't live without the bone marrow. But we know that if we know the cells, if we can target the cells with the specific vulnerabilities and downsize them to a level where the immune system can act. We know, for example, that we have cured millions of people with allogeneic stem cell transplantation. And that is due to the immune effect. It wouldn't do with just the chemotherapy or the specifics. So we need immunology, we need tumor biology, we need basic competence about how the body, the organ works, and of course about how patients react. So we need to lean back and really see how we can collaborate.
Practically, can you give examples of effective anti-cancer treatments that target the cancer metabolism? And Malene Hillebrandt, I mean, you of course, your research is targeted towards monitoring of the cancer and the metabolism inside of the cells, but you do treat cancer as well, of course. Can you give an example?
Yes, it's not exactly directly the cancer metabolism that is behind the effective treatments in my view, or from my perspective, because I'm within imaging, so it's much more diagnostic. So here we use the cancer metabolism to guide us for giving the correct treatment. So it would not be so easy for me to tell exactly an example of an anti-cancer treatment that target the metabolism.
No, I see. I see.
Yeah,
Does anybody else have an example of that?
I mean, methotrexate is one of the methotrexate.
Yeah.
It's one of the oldest anti-cancer drugs that has been used in clinics, and it targets folate metabolism, and it does have a very good impact on cancer cells because basically it stops the proliferation of cancer cells by blocking the production of nucleotides, which are required for the synthesis of nucleic acids, such as the DNA. So the cell cannot divide because if it doesn't replicate the DNA, it cannot divide into two daughter cells. However, of course, the cancer is not the only thing in our body that proliferates, right? So the immune system proliferates as well, right, from the bone marrow and then to produce.
And cancer stem cells proliferate
can-
very rarely.
The
so they
cancer
are
stem
less-
cell proliferate very rarely, so
They're-
attacking just those cells that are proliferating is not enough,
No.
as Eva was saying before. And of course, we have to improve those treatments. And if I may spend a few words about methotrexate, actually, we have found many folate enzymes that translocate and collaborate in the nucleus with epigenetic readers, so proteins that are able to read the chromatin and interpret the chromatin to tell the cancer cell what to transcribe, right? So if instead of targeting cancer cell with methotrexate, we find a way of targeting the cancer cell by impeding the translocation of those folate enzymes in the nucleus, maybe it can be more efficient than, pan-targeting with something that inhibits the synthesis of DNA, basically.
Next question. What emerging metabolic vulnerabilities do you consider most promising for a therapeutic intervention beyond the traditional glucosis-targeted strategies?
Shall I use this to promote a little bit our research?
You know what? We love promotions up here. Go ahead, Mr. Nagy.
So I believe that what I just mentioned previously, the transsulfuration pathway, will be a nice player in this game because it seems that it is quite important in many cellular functions that are related to tumor progression. And we have now a couple of spin-off companies which are dedicated to develop new drugs in this sense, utilizing what we learned about functions and roles of the transsulfuration pathway in drug development in very aggressive tumor types like pancreatic tumor or melanoma. Those are the two which are most advanced in our hands. But please allow me to go back to the house building exercise a little bit, because I think there is one concept that we really need to mention here. I had the privilege to see the development of the cancer mission from the very early days.
Oh.
And the early days concepts, and I think still today, the most important concept is to implement the research findings into the clinic. And when you consider that in terms of drug development, then you have to think about the innovation chain, which is a very complex and very lengthy process. And I have to say that some countries in Europe are traditionally very strong in building an ecosystem which is fantastic for the innovation chain, like the Nordic countries, for example. But the country where I'm coming from and the region where I'm coming from, the Central Eastern European region, is not specifically strong in this,
Why not?
in this aspect. Yeah, that could be another panel. But
Let's not go that route.
yeah. I mean, we have very strong fundamental research. We did actually some research on this topic, why Central and Eastern Europe was not very strong in developing new drugs. We did that as part of the Uncanny U project. I led the work package which was dedicated to inequalities in that one. And it turned out that the technology transfer is what is missing badly from our areas, and funding related to early drug development and the worst. So basic research is there, it's very strong, but the mindset needs to be shifted in some of the researchers to try to do practical, not blue sky.
It
type
seems
of
like it's
research
quite a
activities.
universal problem, though, because this has been touched before
That's right.
on this day, right?
But I just came back from China, where they do it really, really well now, and the speed.
Coming from almost nothing,
The
right?
speed they manage to implement innovation in their ecosystem is just mind-blowing, I have to say. So,
Well, when they set their minds to something, they kick it off.
so this is a momentum for Europe to catch, I think, in that regard.
It's a funding discussion, and it's relevant, but it will take us into another panel as well. Did you want to add something
Yeah, I
to
wanted
this?
to comment, you said, on the Nordic countries, because that is really important for researchers. I mean, in Sweden at least, the researcher, not every postdoc, but the independent researcher has the right to IP.
That's right.
Instead of that belonging to the university. And this is really fostering a wish to take your basic findings forward.
Sweden is the only country in Europe who does that, unfortunately.
Yes.
Yeah.
You can, you're welcome. I'm just joking. No, but the thing is that is really important. I mean, of course, you have to share, you have to finance your research, but it doesn't belong to to the university or to the government. And the university encourages researchers to, for example, apply for IP or patent things before you publish it. So that is actually sort of encouraged. I think that's very good.
Thank you. Hildebrand?
Yes, so no doubt that we have this translational cleft and that it's important to address it, but also after translation, I think in the research that we do is also that you need the different phases of the clinical trials before, and that can also be published, but then you also have the gap from having the evidence to also have the clinical application. I think that's another gap that's also necessary to address.
Okay, let's take the questions here because they're good questions. Is the Warburg effect an adaptation to low oxygen availability, or does it occur even with adequate oxygen supply and result from something else? That's straight into the basic science of yours.
Yeah.
Is that you, Dr. Sedic?
So yeah, it does happen also when oxygen is available.
Yeah.
And actually, it has been a debate for 102 years of why the cancer cell decide to opt for performing the Warburg effect, which basically means stopping production of, let's say, ATP before this pathway goes into the mitochondria, right? And the debate is because the Warburg effect is not ATP production efficient, so the energy that a cancer cell would produce by blocking this pathway is much less than what it would produce if it would continue with the respiration, right, within the mitochondria. And I think there is no answer so we have been speculating for a very long time now that the cancer cell does this because it's much faster, and even though it's not energy efficient, it allows the cancer cell to rewire metabolism in a way that it would produce the building blocks in other ways, right? But I think actually this goes into Peter's expertise probably in the redox metabolism inside the cell and how this rewiring of metabolism allow the cancer cell to be better in terms of redox balance and to then keep going with high proliferation speed and so on. For those that are not stem cells, for those that are really the cancer proliferating cells. So yes, the Warburg effect happens even if mitochondria are working or potentially can work, even if there is oxygen, is really a rewriting that happens in proliferating cancer cells.
Okay, another question it's related to something we talked about some minutes ago. What emerging metabolic vulnerabilities do you consider most promising for therapeutic intervention beyond the traditional glycosis-targeted strategy? So this is a little bit related to what we talked about 3-4 questions ago. What emerging metabolic vulnerabilities do you consider most promising for therapeutic intervention?
I can say something that I think is important, at least for some cancer types, and that is that the cell is very dependent on being able to handle proteins, intracellular proteins, and the proteasome machinery in a cell, that is what really degrades proteins and cleans up, is kind of the local cleaning company. and they are now already targets for the therapy, for example, in myeloma. And cells who have a lot of aberrant proteins, they are more dependent on the proteasome than others. And I think that is something that we can probably expand and use and then combine with other treatment. So that's one example.
Anyone else?
Mitochondrial metabolic pathways, I think, are quite important here.
Yeah.
Because those are the powerhouses of the cell, as we heard previously. And for example, the mitochondrial electron transport chain, I think, is an interesting and important one that can contribute immensely to tumor progression. And it's been researched very actively by a really large number of research groups coming from different areas, and interesting new targets and new treatment possibilities are emerging as we speak. I think that's an interesting area to watch out for.
I want to say I'm going to let this panel run a little longer than according to the agenda, because we were quite delayed due to our little technical problems. So I'm going to let it run just a little further than what's on the agenda for today. And also because you guys are asking really good questions. I want to get back to the protection of findings with IP, which is another question, but I want to get to the AI-driven metabolic modeling becoming a practical tool in designing personalized metabolic therapies. Are any of you working with this already?
Well, I think one interesting thing, and then I pass the word on here, is to mention is that in most cases, when you have a metabolic rewiring within the cancer cell, it does not impact one metabolic pathway. They have a very immediate and harsh effect on each other, these metabolic pathways. So you have to understand how they contribute to each other's activities and what is the bottleneck in that particular cancer cell that you're trying to target. So AI here, I think, will have a major role in figuring out what is most important and what is going to be the most efficient way to come in with.
Did you say will be or won't be?
Will be.
Will be. So the answer is yes.
It is already. Actually.
It is already. Yeah. Do you guys already work with us?
We do use AI for a different purpose, not for
But
modeling.
not for
But
this.
I agree with Peter and what he said, that this is for sure, I mean, it's something that is being used already. What is important, though, is what data we feed the model, right? Because, again, from my personal perspective, if you consider the cell like, like a bubble where metabolic reaction happens, randomly, then it becomes difficult to then discriminate how the rewiring is going on. We really have to take into account the compartmentalization of the cell and where those metabolic reactions are taking place and why they are taking place. Are they needed to reshape the chromatin environment and how the cell interprets the information that comes from the DNA, or are they the result of those, metabolic rewiring, chromatin rewiring, that then affect the behavior of the cancer cell?
What key immune tumor interaction is missing in PDX and syngeneic models that make them poor predictors of immunotherapy response? So we're back to the topic of one of the previous panels.
I can start, perhaps.
Yes.
I think that in real life you have a tumor. That tumor is not only tumor cells, it's also infiltrated by immune cells of various sorts. Some of them would like to counteract the proper immune cells that come from the body. But you also have other types of infiltration. For example, I suppose you've heard about clonal hematopoiesis. That means that in 10% of people 70 plus, you have actually mutated, not very dangerous, but mutated cells, immune cells. And it's a recent paper showing that if you have these in lung cancer, the survival of these patients will be poorer. So in real life, you have a much, much more complicated model. And you have protective immune cells, and you have counteracting immune cells. You have macrophages, a very, very tough cell that have a tendency to work in many ways, both protecting tumor stem cells and sometimes helping out. So we have to make very much better models. We are simplifying things, and I don't think we will get the answers then.
if I may say a few words to the lay audience on this regard, because this question was coming from an expert most likely. PDX is patient-derived xenograft. So what we do in the laboratory when we have a discovery in a cellular system, we try to understand whether this actually is important in vivo, in animals. And the classical animal model in many laboratories is xenografts. Xenografts mean that you have animals with immune-compromised immune deficiency, and you inject tumor cells to these animals, and the tumor develops. But you have to have very little immune system in these animals in order to be able to grow these tumors. Patient-derived xenografts means it's a new generation of xenografts where the tumor from the patient is implanted in these mice, so the heterogeneity of the tumor remains. It's a better model system, but still you need to use animals which have a compromised immune system in order to be able to grow these tumors inside these animals. So for that reason, it was asked how it compares to real life when you actually have an immune system. And this is a very good question, because we struggle with this. In real life, as was said previously, tumor cells are infiltrated by immune cells. So they have a different metabolic profile because of that. It comes back to metabolism, of course, because that's the topic of our discussion. So we always need better and better model systems. And this is why the innovation chain, to come back to that aspect as well, is so long, because after you have something working in vivo models, you have to show that it's non-toxic and then try it in humans. And in a number of cases, even though it's working in animals, it will not work in humans because of many reasons, and this is one of them.
One of the limitations, if I can add something, because I think in the question we also had syngeneic models in which you really take into account the immune system, because you're using cells that are coming from the animal. So the animal in that case that you use in the laboratory has an immune system, and then you can monitor how the cells infiltrate the tumor and how the tumor responds to the treatment and so on and so forth. However, if you think about when humans develop cancer, usually it is not in the young age, right? While the mice that we use for monitoring tumor growth in the lab are actually young mice. So we are, right? So we are missing a component there, which is the systemic component of the body, right? Of the aging body over time.
Mice.
Exactly. So it is, of course, more convenient, let's say, to use mice that are not old to do this type of experiments, right? Because otherwise you should should get them older and then do the experiment, and that would require a lot of time. Plus, they are more fragile and everything, right? There are ethical rules to follow and everything. Sure. But I think it's something that we also need to take into account, right, when we want to propose then the treatment for the patient, because we are not going to treat young people most of the time. We are going to treat people that are already getting older.
Should we focus more therapeutically on enhancing normal cell turnover or cell death in cancer cells instead of inhibiting cell growth?
I can probably, since I started it off, I think yes, but not enhancing the normal cell turnover, rather trying to remove the factors produced by the cancer cells that inhibit it. So I don't think there's possibility, and it would sound a bit dangerous to enhance normal cell turnover, but cancer cells produce a lot of nasty things, cytokines, GDF15, IL-18, a lot of things that make the healthy part of the tissue actually produce less cell turnover. So I think we need to understand the bone marrow microenvironment.
This is actually an approach in redox biology as well, currently, because in many cancer cells we know that these oxidants are overproduced, and for that reason, to keep balance, they also overproduce their antioxidant machinery. So many researchers are looking at how to inhibit those antioxidant functions for the oxidants to overcome their effects and thereby kill cancer cells. And a very sexy area now in redox biology is called the ferroptosis area, where oxidant-induced iron-related cell death is a major pathway that is pursued, how to utilize this to kill cancer cells.
So, last question, and I saved this for last because I think we should not dive too deeply into this. This is on whether you're able to protect your findings with IP and how your therapy will be manufactured. And the reason I don't want to dive too deep into this is because I don't really know if somebody is a potential investor or this is a peer who's asking or what's going on here. So, and it actually matters. So let's just take this question for what it is. Are you able to protect your findings with IP? We already touched a little bit on it. The rules for these things or the way to approach it are
If you're in Sweden,
different in
yes, you
Sweden.
can. Yes.
The answer will be
Yes.
yes. Otherwise,
Yeah, so if you
normal rules apply, right?
Yeah, exactly. I think every country probably has
Yeah.
their own rules. In Spain you can, but it's shared with the institutes. It's not just for the researcher, right?
And the reason I am speculating whether this is a potential investor or not is because of this follow-up. How are you going to manufacture it? Because, of course, that requires...
Well, let me turn it around. I think you must protect your discoveries by IP.
Yes.
Otherwise, you're wasting taxpayers' money. Because if you publish in Nature, you publish your new target in Nature, and if it's not protected by IP, no big pharma will be interested in that discovery anymore. So what you do is actually you wasted a discovery which was provided to you by Nature.
Oh, good point.
And you cannot utilize this for treatment anymore because big pharma will never pick it up. So you have to protect your IP. It's a big struggle for everyone.
Back to the
We
collaboration
do
with the
struggle,
industry, right?
and it costs a lot,
Yeah.
but it's mandatory.
Right. Okay, that was the short answer I was looking for this last one. I cannot thank you enough for both answering all these extremely good questions and also, just on my own behalf, thank you for being so metaphorical in your answers so that even I could understand. I love that, and I love that there is a sexy research and there is a little house and there's a little factory inside of it. Thank you for that. That was full of surplus, and thank you.
Thank you.
So we need a cup of coffee now, and I am going to ask you to be back here at 4. Again, I just nipped and tucked a little bit, and we'll be nipping and tucking, and we will end as planned. But we'll nip and tuck the next panel a little bit, and 4 o'clock, 25 minutes will be good for a cup of coffee.